Okay, let's start today's class. I'm going to share the screen and let me know when you can see the screen and the PowerPoint. Can you see the PowerPoint?
Yes, sir.
Oh, good. So I'm trying to go to full screen mode. I think now I'm in full screen mode. Okay. Sorry. So today we will be finishing the antigen part. We have a little bit part remaining, and then we'll switch to other things. First, we'll talk about this epitope. And what is epitope? Well, when I'm talking about antigen, or rather we should say immunogen, you can imagine one bacteria maybe is an immunogen. Now, our immune cells, such as B cell or T cell, they do not interact, or they do not have the capability to recognize the whole bacteria, the whole virus, the whole fungus, entire molecule. Because you understand that for a B cell or T cell, they do not have any understanding about what is bacteria, what is virus. They do not have any brain or anything like that. So what they're recognizing only certain molecules that is present on their surface of the antigen or surface of the immunogen. Okay. So now, if you have a little bit of understanding, they will be able to understand that a bacteria on its surface can have hundreds of different types of protein, hundreds of different types of glycoprotein, that is protein plus carbohydrate, lipid molecules, etc. So only few of them are getting recognized by these immune cells, that could be B cell or T cell or other type of immune cells. So only those sites present on the bacterial or immunogen surface which are being recognized by our B cell or T cell type of immune cells are known as epitope. Okay, so the entire thing is not important over here. Only the epitope site is important. So immune cells do not interact with or recognize an entire immunogen molecule. Instead, lymphocytes recognize only the discrete sites on the macromolecule. So some amino acid, some of the carbohydrate molecules, those are known as epitopes. In other, they have also another name that is called antigenic determinant. So epitopes are present on the antigen or the immunogen. Okay, some sites. So we can say, we can define it classically, that epitopes are immunologically active regions of an immunogen that bind specific antigen-specific membrane receptor on lymphocytes to secrete antibody. How is so? So the definition may seem a little bit complicated. So if you take this example, so here we are seeing a B cell. It has membrane-bound receptors. Okay, and now this is our immunogen, a bacterium, and it has one epitope here. Okay, so this is the epitope site that is being recognized by the B cell receptor. So epitopes are immunologically active regions. So this is the active regions of an immunogen. So epitopes are part of the immunogen that bind antigen specific membrane receptor. So this is our antigen specific membrane receptor, or present in where? On the lymphocyte, that is a B cell or B lymphocyte. Okay, or to secrete antibodies. That's the secondary part, because you already know what is going to happen next. This B cell will internalize the antigen, will present it to the T cell or T helper cell, and then finally antibodies will be produced. So this is the epitope site, immunologically active region on immunogen that bind to antigen specific membrane receptor on lymphocytes. Okay, now if you think about the lymphocytes, mostly you have two lymphocytes, very important, B and T, but they do not have, they have different epitopes to bind. So whatever the epitopes we're seeing here is bound by B cell membrane bound receptor that will not be recognized by any T cell. T cell will recognize something else, maybe it's present in here, that will be recognized. So when this B cell will present this antigen to the T cell, if the B cell present this particular epitope to the T cell along with the MHC molecule, T cell will not be able to recognize that through its membrane bound TCR or T cell receptor. They require different types of epitopes. So this bacterium, the entire one, will be internalized, will be fragmented into small pieces, and now all those pieces will be presented to the T cell, and T cell will recognize one particular epitope. So you can chop it in 100 fragments, you can present it one by one, T cell will reject most of them, and then it will recognize only one particular epitope that is present over here. So when that fragment is presented, then only TCR will bound to it and will be activated. So B and T, both cells have different epitopes to recognize on the same antigenic molecule, like a bacteria, one particular bacteria. Okay. So you understood what is epitope and the peculiarity that B and T cell have different epitopes to bind. Okay, so this is another, this is just a graphical representation of an epitope. So once again, you can consider that this is a bacterium and we are seeing it has 3 epitopes. So there, theoretically, there could be numerous number of epitopes or there could be one epitope. But most of the time we found that the number of epitopes present on a antigen is more than 1. So let's see for this example that 3. And why we're giving it different structures? We're trying to say that these epitopes, mostly they're protein, their 3D structure is also different. So one particular antibody will not be able to bound to all 3 different types of antibody. So you see, whatever antibody you'll be getting will be epitope specific actually. A single antigen may possess several different epitopes and different antibodies may bind to different epitopes on the same antigen. In this picture, if you see, the same bacterium we're showing here with 3 different epitopes. One is a brown color triangular shape, one is yellow round shape, another is the green rectangular shape. Okay. Now you are also seeing 3 different types of antibody matching with the color. So first one, the green color, you see, it has a rectangular shape of epitope and our antibody also has a rectangular shape. Okay, so it will be easily able to bind to this. So this green one is actually specific to this epitope only. It has nothing to do with the other epitopes. Similarly, for this one, the cream color or brown color one, it can see its binding site is triangular type of cells. So it's compatible to bind only to this triangular epitope. It will not be able to bind to the others. So this antibody is specific for this particular epitope. Similarly, the yellow one, its binding site is roundish in shape and it can easily bound only to the round shaped epitope, not others. So each and every epitope, for each and every epitope, we have different antibodies. Okay, that's why, and now you already know that the source of this antibody is actually one particular type of B cell. That means from green, this antibody is coming from one B cell, this brown color is coming from another B cell, and this yellow one is coming from another B cell. Okay, so that's why. So if you inject some antigen to an animal, say rabbit or mice, and after 1 month or after that, if you bled the animal and try to collect the antibody, what we'll be getting always is a mixture of antibody, brown one plus the green one plus the yellow one. Just for the example, I'm taking that. It could be more than 3, it could be 2, something like that. So mixture of antibody. That's why is called polyclonal antibody. That means the antibodies are coming from different clones of B cell. Poly means many, different clones of B cell. Number of antibody and number of B cell is the same. That's why it's called a polyclonal antibody. Okay. So a single antigen, the main thing here, possesses several different epitopes, and different antibodies may bind to different epitopes on the same antigen. Okay. Now, how they bind, just for information, antibody binds to the epitope by weak non-covalent interaction. We know there are a lot of two different types of major bonds are there. One is weak interaction, another is a strong interaction. So strong interaction or covalent interaction takes place only when there's a sharing of electrons up there. Okay. But this type of interaction, antibody and epitope, does not involve any electron sharing. So how they're binding? They're binding through weak non-covalent interactions, such as hydrogen bond, okay, ionic interaction, van der Waals interactions, etc. Always weak. You may have observed that in case of biology, weak interaction always the major force. Okay, strong interaction is seldom found. Weak interaction, but they found in a huge number, so that's a different issue. Okay, so antibody binds to the epitope by weak non-covalent interaction. And one thing is that as the interaction is non-covalent, is weak in nature, operate only over short distance. That means if this is our antigen, and if this we consider an epitope site, and if we have the antibody at, say, this distance, okay, so this distance might not be good enough for the antibody to bind to this epitope. That could be too far away. The antibody is too far away from the epitope site. Weak or non-covalent interaction only operates over a short distance. So this will be able to bind to this epitope only when this antibody will come into very close to this epitope site. So whenever we try to do any experiment in the laboratory involving antigen and its corresponding antibody, we have to necessarily keep them very close to each other. Okay, we can take a petri plate, we can form a jelly-like substance called gel, and then we can make some well using a cork borer, and in one well we can put antigen, and we can make another well where we can put antibody. We'll try to discuss this some later point of time if we got the time. So then only you can expect that these two will react with each other because they're in a very close proximity. But if you make this well far away in the plate like here, you cannot expect to see a very good interaction between them because the epitopes and the antibodies, that interaction is weak, non-covalent type. That's why they operate only over short distance. For a strong bond, the antibody's binding site and epitope must have complementary shapes that place the interacting groups near each other. So now the thing is that as the bond is weak in nature, how they will maintain a strong interaction between them. Okay, that's why the thing of complementarity comes, complementary shape. Okay, that's why I was showing here that for rectangular epitope, we have rectangular type of antibody binding site, complementary shape. If it is a round disk, we have round disk complementary antibody site. If it is a triangular, we have triangular sites. So that's complementarity gives the strength of binding between an epitope and an antibody thing. Okay, so this requirement poses some restriction on the properties of epitope. What requirement? We said that epitope and antibody must be coming into very close to each other. They must be complementary to each other. Their chemical groups should be come into close contact. But that makes a little bit problematic in some time. The size of the epitope that is being recognized by the B cell can be no larger than the size of the antibody binding site. If we take this example once again, okay. So this is our antigen, bacteria maybe. We have three different epitopes which will bind to its corresponding antibody with a complementary cell. Okay. Now, say this particular antibody supposed to bind to this particular epitope supposed to bind to an antibody which is complementary round this type of shape like this. Okay. But if somehow this epitope is large like this, okay, and our antibody is like this, it's round, but still it cannot bind to it exactly. It can complementarity is not being maintained because the epitope is huge in size in comparison to our antibody. So even though the antibody has same shape, round, but cannot accommodate the whole epitope, so that will be a problem. The binding between epitope and antibody will not be good enough. So that's why size of the epitope that is being recognized by the B cell. So if we see this is a bacterial epitope being recognized, can be no larger than the size of the antibody binding site. Epitope always must be equal to the size of the antibody at the max or less than the size of the antibody binding site. That is the condition over here. Size of the epitope recognized by B cell can be no larger than the size of the antibody binding site. And the reason I just have explained. Okay. Now, for any given antigen-antibody reaction, so you understand that in our system, immune system, always antigen and antibody are reacting to each other. So that given any antibody-antigen reaction, the shape of the epitope that can be recognized by the antibody is determined by the shape assumed by the sequence of amino acid in the binding site and the chemical environment they produce. Okay. So here, once again, if you look into this picture, how this size is being determined, how it is becoming rectangular, how it becoming triangular or around this. That shape, it's not size, the shape is determined by the amino acid sequence. Antibodies are basically protein or glycoprotein. That means their main constituent is amino acid. So how the amino acids, what type of amino acids are there, how many number of amino acids are there, those things, as well as the environment that has been mentioned over here, chemical environment, that means the pH, the temperature, etc. Those things determine that what type of shape they will take in a three-dimensional space. Will it be triangular? Will it be rectangular? Will it be roundish or not? Okay. So two things determining the shape of the antibody when antibody is reacting to antigen. One is the sequence of amino acid in the binding site, not in the whole antibody. Sequence of amino acid in the binding site of the antibody and the chemical environment at which or that they produce the antibody. Chemical environment means temperature, pH, osmolality, ionic strength, etc. Okay. So this is just another picture. So you can think that this is an antigen. We have 4 different epitopes are there, different shapes. You can see one type of shape over here, another shape like here. This is another shape, and this is another shape. Okay, and 4 different antibodies and 4 different colors are binding to these epitopes. 1, 2, 3, 4. So you can say these are the epitopes and 4 different types of antibodies, each and every one specific. So yellow one specific to this one. So this magenta one specific to this one. This violet one is specific to this one, and this green one is specific to this one. Is specific. Okay. So this is about the epitopes. So once again, epitopes, immune cell do not interact with or do not recognize an entire immunogen molecule. Instead, the lymphocytes like B cell and T cell only recognize small discrete sites on the macromolecule, that means protein, carbohydrate, fat, etc., called epitopes and also called antigenic determinant. Okay, B and T cell recognize different epitopes on the same antigenic molecule. A single antigen may have one, most of the time more than one type of epitopes, and different antibodies bind to the different epitopes. binding between the epitope and antibody is determined by weak non-covalent interaction. And that's why it is very necessary that they operate only over a very short distance. Okay? And for establishing a strong bond by using a weak force, it is very much necessary that the epitope and the antibody must have complementary shape. Okay? To fulfill those conditions, two things are coming over here. One, the size of the epitopes that is being recognized by the lymphocytes cannot be larger than the size of the antibody. Number two, how the antibody is getting complementary sites according to the epitope shape, complementary shape according to the epitope shape. That is determined by the sequence of amino acid present in the binding site of the antibody and as well as the chemical environment that they produce. Okay, so that is the first remaining part I had about the epitope. So I will be stopping at this point before I go to the antibody thing, and I will be providing you the attendance sheet at this point so that you can complete the attendance thing, and then I'll be back. We'll start the antibody. Okay. So here is the link, the chat box. Open it and confirm that you can provide the attendance. Is it opening? Is there any problem?
Yes.
Okay, fine.
Yes, sir, it's open.
Yes, sir, it
Good.
is open.
Very good. I'll be back in say 3 to 4 minutes time. Okay, I'm back. So I have shared the screen and the PowerPoint. I hope you can see it. So now we will switch to other thing, a new thing that is antibodies. So far we're talking about the antigen. So now we'll talk about or try to understand the antibody. The first we'll talk about an experiment. I think I mentioned about this experiment in the first class or so, something like that. Here we'll try to understand the discovery of antibody thing in a more detailed way. So that was a very classic experiment done in 1939 by Tiselius and Kabat, two scientists. Okay, what they did. So now you have a pretty much, I hope, basic idea, but then too you'll be able to understand the thing. So first, they took a protein which is known as ovalbumin. Ovalbumin protein is very much found in the egg. Okay, so this ovalbumin protein, they injected or immunized rabbits with this protein. Now, ovalbumin protein is a foreign thing for a rabbit. So very naturally, rabbit's immune system will be able to recognize, or rabbit lymphocyte will be recognized the ovalbumin as a foreign protein. And what is expected that they will produce anti-ovalbumin antibody. So in generally, we use this sign to say anti. Okay. So anti-ovalbumin antibody will be produced. An antibody, we use a abbreviation capital A, small p. Fine. So now what they did, Tiselius and Kabat, after some time, they take the blood sample, bled the animal, okay, and divided that, and then isolated the serum. Of course, you know the serum, and then divided the serum into two parts. One is part A, and another one is part B. Very good. Okay. So in both cases, part A and part B, there is coming from the same serum, and we can expect that both part A and part B should have this anti-ovalbumin antibody. Fine. Then they took just the part A, kept aside the part B, and electrophoresis, they done the electrophoresis of part A. Electrophoresis is a very common, commonly used or frequently used technique in biotechnology or immunology. Here we are doing here that you are trying to separate different molecules present in a sample using an electrical force. So here, what we use agarose or agar to make a jelly-like substance. You can imagine that you have a jelly-like substance which will be solid, liquid at first, and then will be solidified. So this is your container or vial container, and you are pouring in the jelly-like substances, and it is getting solidified after some time. And then you are making a groove or a well at one end, and you're putting your sample over there. That sample could have been mixture of 3, 4, 5, 6, 10 different things. And then you're using an electrical force. Okay. By the term of electrical force, these molecules will be running from the cathode to anode. Okay. They are all basically negatively charged. So they will be attracted to the positive electrode. But the thing is here, as they all are experiencing the same electrical charge, but as I said, this mixture of sample could have, say, 1, 2, 3, 4 different type of samples, different types of compounds are there. All of them are not equal molecular weight. Their molecular weights are different. Some are very light, some are moderately heavy, some are very heavy. So if all of them are experiencing same molecular electrical force, the lighter one or lightest one will be able to reach to the positive electrode first. One, then the little bit of more heavier than that, then the moderately heavier, and the heaviest one will be lagging behind. So 4 different people you can think is running for a short distance, and some of the boys are very thin and very agile. He or she can fast, and others are maybe a little bulkier, heavier in their appearance and cannot run that fast. So it will be lagging behind. So finally, you'll be able to see this type of separation. You'll be able to see, okay, now I understand that this sample is containing 4 different types of molecules are there, a mixture of 4 different types of compounds. Okay, and you can take one of them each out and do your subsequent study. So the same thing also done here by the Tiselius and Kabat with the part 1. part A of the serum, which is containing the anti-ovalbumin antibody, hypothetically. So when he got this type of result, there is no relation with this color. No color is there. This is just an artist impression, you can see. So he also got this type of 4 bands, which says bands. Okay. So later he did all the biophysical studies and he found that these 3, green and lighter green and even more lighter one, these 3 can be categorized as a globulin protein. And he named them as alpha, beta, and gamma globulin. And another one he got that is called ovalbumin. Okay, ovalbumin protein. So 1 ovalbumin and 3 types of globulin, alpha, beta, and gamma. So electrophoresis of the one part, that is part A, is done and revealed 4 peaks corresponding to albumin and the alpha, beta, and gamma globulin like this. Okay, just think about the blue line over here, not the black one. So the blue line shows the height of the peak. Height of the peak is showing the abundance. That means how much amount of one particular protein is there. So albumin is the majority. Maybe albumin heavier, maybe albumin is a very lighter one here as you can see from this picture. But albumin is much abundant or dominant type of molecule that is found in the serum. That's why its peak height is so much. Okay. Whereas the globulin, alpha globulin, beta globulin is less. Okay. But another one, gamma globulin, like this one, is also giving a good peak height. That means the second abundant molecule is gamma globulin. Most prevalent or dominant one is albumin. Second one is gamma globulin. And the third and fourth one is alpha and beta globulin. That they found by doing the electrophoresis of the one part of the serum that they collected after immunizing the rabbit with ovalbumin. Fantastic. Now they concentrate on the part B. Part B also the same thing, just like part A. But here they did a trick. What trick? They added ovalbumin, the same ovalbumin that they used to immunize the rabbit, the same ovalbumin they used here to react with the part B of the immunized serum. Okay, that was containing hypothetically anti-ovalbumin antibody. So anti-ovalbumin antibody was there, but you are also now adding the ovalbumin against which you have raised this antibody. So ovalbumin is actually the antigen that was primarily the first phase was used to inject the rabbit. So now what you have? You have ovalbumin as an antigen and anti-ovalbumin as an antibody. So when they're coming into close contact here, they will react. Then what will happen? There will be an antigen antibody complex will be formed. Ovalbumin plus anti-ovalbumin antibody complex. When the complex will be formed, they will precipitate at the bottom of the tube. After some time, so 1 hour, 2 hours or so, or sometime maybe overnight, they will react. They will become a big complex, and due to the gravitational force, they will settle down at the bottom. Okay, fine. So that settle down. Then Thesalius and Kabak take out only the liquid portion that didn't settle down at the bottom, remaining serum protein. So now we are confident more or less that this serum protein is not containing any anti-ovalbumin antibody, because whatever anti-ovalbumin antibody was present in the part B already got complexed over here and precipitated in this tube. So there might be very less or nothing in the supernatant or the liquid portion. Okay. So then what they did again the electrophoresis this part, just like what they did in the part A. So only difference is that only the ovalbumin antibody has been complexed and removed from the liquid. So when the electrophoresis did the electrophoresis and use the plot, so now you can see two different plots carefully. One is the blue one that we have already seen, that high maximum number of albumin. Second maximum is gamma globulin, and beta and alpha are very less in number. So now the black one is coming from the part B. Part 1, part A is coming, blue line is from part A, and the black one is coming from the part B. So what you're seeing here, in case of part B, albumin protein is still there, no issue. The same is still showing the same abundance. Alpha and beta globulin also showing the same amount of abundance like part A. Only dramatic change has happened in case of the gamma globulin. Part A, you are seeing such a high peak of gamma globulin, but for part B, you are getting a very less peak, very small peak, comparable only to the alpha and beta globulin. Okay, so this is, we have seen the removed by antigen. Already you have removed the antibody from the system. We have already removed the system. So this is giving, so what does it signify? It clearly signifies that what is reacting here as antibody with the ovalbumin antigen, what is reacting in the part B with the ovalbumin antigen is nothing but gamma globulin. That's why you are not getting much gamma globulin in the part B after antigen antibody has been complexed and precipitated in the tube. Okay, so a comparison of these two serum aliquots revealed that there was a significant drop in the gamma globulin peak in the aliquot that had been reacted with the antigen, that has been shown here in the black line. So that was a very classic experiment done by Tiselius and Kabat in 1939, and thereby they confirmed that the gamma globulin fraction is actually the part of the serum which contains the antibody, okay, later which were called as immunoglobulin, as it has the role, very significant role in immunology. So gamma globulin is the fraction of the serum which is containing the antibody. Other part of the serum, alpha, beta globulin or albumin, are not the immunoglobulin. Later, this theory will change a little bit. Later, people found that the alpha beta globulin also contributes a bit, but not as important as the gamma globulin. Later, we'll see, we'll come into this within a few minutes maybe, that later this gamma globulin got the name immunoglobulin G or IgG. We always write them like this: I for immuno, small g for immunoglobulin, and then capital G to denote one particular class. So this is the main class of antibody molecule, is indeed mostly found in the gamma globulin fraction. Significant amounts of it and other important class of antibody molecules. We will see there are 5 different class of antibody. Those are also found in the alpha and beta fractions. Okay, but that is the different story. But main issue is how it has been known that which part of the serum is containing the antibody. That, thanks to the Tiselius and Kabat's experiment, this way we found that the gamma globulin fraction is the main thing. So now we will talk about the structure of antibody, whether antibody has a fixed or specific structure, or is there any peculiarities about the structure or not. So what are the basics? Actually, the structure of antibody is quite complex in nature, so we will not go into that. We will stick ourselves to some basic features of the antibody molecule. Okay, so this is a classical shape of antibody molecule, classical structural shape, kind of a Y, okay, but it has a double legs over here. So antibody molecules have a common structure of 4 peptide chains: 1, 2, 3, and 4. Peptide. Peptide means small protein. Okay. So 4 peptide chain that constitutes an antibody structure. Okay. Now out of these 4, two of them we will be we will categorize, we will classify them as a light chain and or L, and two of them as a heavy chain. So this outer two, this one as well as this one, is known as light chain, and this entire this one and this one is known as heavy chain. What is the meaning? The light and heavy means who are lighter or who are heavier in terms of molecular weight. Light chains are actually only 25,000 Dalton in its molecular weight. So this one is 25,000 Dalton. This one is also 25,000 Dalton. Whereas the heavy chains are actually double in the size or weight. They have 50,000 Dalton molecular weight. Okay. So two light chain, 25,000 Dalton, 25,000 Dalton, total 50,000 Dalton, and two heavy chains, each of them 50,000 Dalton, 50,000 Dalton, total 100,000 Dalton. 100,000 plus 50,000, 150,000. So generally speaking, one antibody molecule is found to be 150,000 Dalton in molecular weight. So four peptide chains, two identical light chain. Maybe I missed the term identical. So this light chain and this light chain are exactly identical. And this heavy chain and this heavy chain is also exactly identical. Okay. Then how they're bound to each other? So they cannot float in the air. Each of the light chain, if you see, each of the light chain like this is bound to the corresponding heavy chain with a bond which is known as disulfide bond. Is also one type of weak interaction. Disulfide bond, when two cysteine amino acid come into close contact, then they form a disulfide bond by the process of oxidation. Okay, so light chains is bound to the heavy chain by disulfide bond to form a heterodimer known as HL. So hetero means different and dimer means and dimer means two different types of two molecules. So you can see there cannot be said homodimer. Homo means the same because it's a light chain and the heavy chain, two different types of molecule. One is different in its protein sequence, amino acid sequence. Another is also totally different. They're different in terms of molecular weight as well. So these two is forming a dimer. One light chain and one heavy chain is forming a dimer, and the dimer is HL, heavy light. Okay, and each heavy light dimer, you will find the light chain is bound to the heavy chain by disulfide bond. Similarly, here also one light chain is bound to its corresponding heavy chain by a disulfide bond and forming another heterodimer. So you can see actually antibody is a dimer of heterodimers. One heterodimer over here and another heterodimer over here. And then these two are coming together and forming a dimer. So last question remains that now how these two heterodimers are bound to each other. These two heterodimers once again is also bound to each other. I mean, two heavy chains are bound to each other by disulfide bond. Only difference is that the number of disulfide bond is 2, not 1. So here one disulfide bond, here one disulfide bond when the heavy chain and light chain is binding to each other. But in case of intra-heavy chain bonding, we are getting 2 disulfide bond. So similar non-covalent interactions and disulfide bridge link the two identical heavy and light chain combination or the dimer. As I said, one dimer is over here and another dimer is over here, and these two heterodimers are getting linked with the disulfide bridge. So you can call them a dimer of dimers. One dimer, two dimer, and the dimer of dimers, the whole thing. Okay, that is a very basic thing about the structure. Now, if we go into a little bit of deep, then found that 110 about 110 amino acids of the amino terminal region of a light or heavy chain varies greatly among antibodies of different specificity. We know by theory, already discussed, that there is no uncountable number of antibodies is possible. As I said, that we have the potential to produce antibody if something and some bacteria, some other organism comes from the Mars today or from the moon or from other planet. Okay, so that means huge versatility and they are very specific. Now you are showing a simple basic structure, then how they will be so versatile and specific. Now what is the meaning of N-terminal? Actually, when we talk about the protein, we'll define the protein as a two different end. One is N-terminal or amino terminal, and another is C or carboxy terminal. So, of course, I'm not going into the detail, but as this is a protein, okay, a peptide, that means if you consider this light chain, this is the N-terminal of the light chain, and this is the C. For heavy chain, this is the N-terminal, and then goes, and sorry, totally wrong. So they have drawn it a little bit different. So this is a light chain, this is the N-terminal, and this is the C-terminal. For the heavy chain, that starts with the N-terminal and then the C-terminal over here. Similarly, also go like that. Now, the first 110 amino acids of this heavy chain and first 110 amino acids of the light chain from its N-terminal, they are found to be varying greatly from one antibody to another. So the specificity of the antibody or the versatility of the antibody depends only on this particular region. Okay, other region remains more or less constant or fixed, only this. So this region forms the actual antigen binding site where our antigen, maybe a bacteria, will bind. So the first 110 or so amino acids of the amino terminal region of a light chain or heavy chain varies greatly among antibodies of different specificity. Okay, this variable segment, that's why it's called V region. So this is also the V of light chain and V of heavy chain. So if it is a V of light chain, then we can call them as VL. If it is a variable region of the heavy chain, we can call them as VH. The same also goes or true for here, VL, like VL and then the VH. But the remaining portion, as I said, doesn't vary. They remain constant for all different types of antibody. That's why the blue marked region, everything over here is known as constant region. But because this is a light chain, we can call them CL or constant light chain region, or we can call them as HL or sorry, CH or constant region of the heavy chain. Variable region and constant region. So what is the difference? Only difference is that this region, the first 110 amino acid, varies from one antibody to another antibody, but the remaining portion, which is known as constant region, they do not vary from one to another. So whatever I mentioned is mentioned over here. Okay. So look into the class structure of the antibody once again. Basic structure: 4 peptide chain, 2 identical light chain, 2 identical heavy chain. Light chain size 25,000 Dalton. Heavy chain each is 50,000 Dalton. So total 150,000 Dalton. One light chain remains bound to another heavy chain by single disulfide bond. And then the heavy chain remains bound to each other by 2 disulfide bond or non-covalent type of interaction. Now, the first and 100, the entire chain, it doesn't behave in a similar way. We can divide them into 2 parts. One is the variable region, another is the constant region. Variable region means the region which is varying, which is from one antibody to another. And where it is found? It is found in the first 110 or so amino acids of the both light chain and the heavy chain. They forms the antigen binding region known as VL and VH. The remaining portion, the major portion, is actually constant. So they are known as Cl or CH. Okay. So this is the very basic structure of antibody. Now I will see the class of antibody. Antibody all are not same. There are different types of classes are there, and their biological activities also differs from each one another. So as I said, that only this region is constant, the variable region always varies. Now, various immunoglobulin isotype or types and classes are found. What is the basis of making them different class? Basis is that this heavy chain constant region, although as I said, the heavy chain constant region remains same for the constant for all the antibodies, but class-wise, they're different. That means for one particular class, we'll be getting one particular heavy chain constant region. Another class will be getting another type of heavy chain constant region. For another class, we'll be getting different type of heavy chain constant region. So among between classes, they remains the same, but interclass, they will be different. So immunoglobulins could be classified into different types or classes based on the fact what type of heavy chain constant region they have. Okay. So we have based on that, we can divide the immunoglobulin into 5 different classes. I was just talking about the IgG. So immunoglobulin G, the one. Number 2, immunoglobulin M. Number 3, immunoglobulin A. Number 4, immunoglobulin D. And number 5, immunoglobulin E. What this big capital letter stands for, GMADE, stands for the type of the heavy chain they are having, heavy chain constant region they are having. Okay. So it is a translation of the Greek word. Say, for example, the immunoglobulin G. For immunoglobulin G, the H chain or heavy chain is gamma type. As they're having the gamma type of heavy chain, we can call them immunoglobulin G because G is the English word for the gamma. Okay. Similarly, for another class of antibody, this heavy chain is mu type. If it's a mu type, then we can call them as immunoglobulin M. For third one, the heavy chain is alpha type. That's why I got the name immunoglobulin A or IgA. For the fourth one, it was delta type. That was got the name immunoglobulin D. And finally, the fifth one, the heavy chain type is epsilon. That's why it's got the name immunoglobulin E. So 5 different immunoglobulin class: IgG, IgM, IgA, IgD, and IgE. What is the basis? Basis is what type of heavy chain constant region they have. They have heavy chain constant region type gamma, mu, alpha, delta, and epsilon respectively. Okay. So we are talking about different types of class of immunoglobulin because their property, their function is way different from each other. Although all of them are antibody, but their biological property or their biological function is not the same. Okay. So we'll focus here on the immunoglobulin G type. This is the most important one because this is the most abundant class. I should not say most important, but is the most abundant class. Okay. In serum. So total serum immunoglobulin, whatever you are getting, 80% is nothing but immunoglobulin G. So only the 20% is being shared by the remaining 4, IgM, IgA, D, and E. 80% is immunoglobulin G containing gamma heavy chain. Okay. So what the immunoglobulin G does, main function, I just said, immunoglobulin is the main type of antibody found in blood and extracellular fluid, allowing us to control infection to body tissues. So whenever we're talking about any antibody, that could be against the COVID or for any other type of disease. So when we're talking colloquially or commonly as antibody, actually we're referring to the immunoglobulin G. Okay. we know immunoglobulin G. So the antibody and immunoglobulin G become a synonymous, but that doesn't mean that all different four types are not important. They are also very important. I will come to those later point. So immunoglobulin G is the main type of antibody found in the extracellular fluid, allowing us to control infection by body tissues. Number two, immunoglobulin G is the only class of immunoglobulin that can cross the placenta in humans and is largely responsible for protection of the newborn during the first month of life. Maybe you know that newborn human children, babies, they cannot produce antibodies on themselves for a few months, 1 to 3 months more or less, or of course nothing in the first months of life. Okay, so that's why they're becoming very susceptible to different types of microbial diseases, contagious diseases or infectious diseases. Okay, so what is the way to protect the human baby? One is that they have some immunoglobulin that they received from their mother when they were in the uterus. Okay, so placenta, maybe you know about the placenta. Placenta is a very thin membrane by which the mother and the baby is separated in the uterus. Okay, so placenta, through the placenta, mother can pass to the nutrients, the oxygen, as well as the immunoglobulin G. So the fetus here is receiving the immunoglobulin G from the mother. And that immunoglobulin G is also protecting the baby at least after the birth, at least for the first month of life. Other type of antibodies, M, A, D, and E, they cannot cross the placenta. Placenta is very much selective about the thing that which particular object will be able to cross or which will not be able to cross. So different other regions are there. That's why other type of immunoglobulin cannot pass. So IgG is the only class which can pass the placenta, and they are largely responsible for the protection of the newborn. There are another type of protection newborn babies that they get it, but I will come to that in the later point. Okay, so immunoglobulin G is a major immunoglobulin found in blood, in the lymph node, even in the cerebrospinal fluid, and also the peritoneal fluid, and key player in the humoral immune response. So more or less, whatever I'm saying in the number 1 point is the same also number 3 point. They are everywhere and they are providing the major protection to us. Okay. Now, IgG is produced in a delayed response to an infection and can be retained in the body for a long time. So IgG is a very efficient, it's omnipresent everywhere, that's okay. But IgG is not gets produced at the initially. It takes some time. There is a delayed response. Okay. So first, when one particular antigen, say a bacteria, enters in our body, our immune system reacts to that because it's an immunogenic and produces antibody. But what type of antibody? What class? Is it IgG? No. IgG is not the first one. IgG comes at the second or third point. IgM comes first, but that we'll talk later. Okay. So there's a delayed response. But on the brighter side is that IgG remains there for a longer period of time. So their shelf life is much long. The longevity in serum makes IgG the most useful for passive immunization by transfer of this antibody. So once again, whatever antibody we're talking about, we're thinking that whether antibody is present in the serum or not after the vaccination against COVID, etc. You must have been seen in the newspaper. People are debating about those things. So we're talking about immunoglobulin G, whether immunoglobulin G is present in the serum for a long time or not for the immunization. Now, our whole concept of this vaccination, whole thing that antibodies will be there for a longer period of time. Antibodies will protect us from that specific disease based on the immunoglobulin G, because this is the antibody which we expect to be there for a longer period of time. Okay, so that is the beauty of immunoglobulin G. Because of its relative abundance and excellent specificity towards the antigen, immunoglobulin G is the principal antibody used in immunological research and diagnostics too. That's why I'm saying that antibody and immunoglobulin G generally become synonymous. That doesn't mean that all 4 different other types of antibodies doesn't have any significance. They have immensely significant, very important jobs to do. Maybe I will talk about that later. But immunoglobulin G is the principal one. They're having the 80% of the serum antibodies, immunoglobulin G. And all our design, our strategy about this vaccination thing is totally based on this immunoglobulin G, at least most of the time. Okay, so now this immunoglobulin G letter could be classified into 4 different type of subclasses. 1, 2, 3, 4. Okay, so that is also based on what type of gamma chain sequence is there. So there is no point in discussing those at this class. Just you can think about that immunoglobulin G is a one type of immunoglobulin that could be further subdivided into 4 types, 1, 2, 3, and 4. Okay, and they have been named as 1, 2, 3, 4 actually based on their relative abundance in the serum. That means you will get the maximum amount of thing is IgG1 with the serum, then IgG2, then IgG3. You will get very little amount of IgG4 in our serum. So 5 different class of antibody and then 4 different class of IgG subclass. If you look into the IgG1, as I'm saying, 60% of the immunoglobulin is nothing but IgG1. They are predominantly responsible for thymus-mediated immune response against protein and polypeptide antigen. IgG2 also has responsible for immune response against carbohydrate polysaccharide. So there is a distinct difference. If the antigen is protein or polypeptide in nature, then IgG1 will be effective. If the antigen is carbohydrate or polysaccharide in nature, mostly IgG2 will be responsible, will be effective. IgG3 that is found even less than 10% plays a major role in the immune response against polypeptide and protein again, but it's not clear what a particular role IgG3 plays or even the IgG4. Very less number. So we're not very clear about IgG3 and IgG4 role, but we know that IgG1 becomes effective when the antigen is polypeptide in nature or protein in nature, and IgG2 becomes effective when the antigen is carbohydrate or polysaccharide in nature. And relative abundance wise, IgG1 is a more than 60% and IgG2 is more than 20%, 20 to 30% range. So IgG containing the 80% of the serum antibody and IgG1 is 60% of the IgG. Okay, so if we look into this function, class and function thing once again, immunoglobulin class, 5 different class, G, M, A, D, and E, based on their heavy chain constant region, gamma, mu, alpha, delta, and epsilon type, and IgG1, the most abundant class. 80% of the serum immunoglobulin is nothing but immunoglobulin G. Functions, they're the main type of antibody found in blood and extracellular fluid, allowing us to control the infection of body tissues. They're found in blood, lymph fluid, cerebrospinal fluid, or peritoneal fluid. And importantly, this is the only class of immunoglobulin that can cross the placenta in humans and protect the fetus from any type of infection, and also largely responsible for the protection of the baby after their birth, at least in the first 1 or 2 months in their life. And we have found that IgG is the produce a delayed response. They do not found in a quick response, a delayed response, but the good thing is that they retained in our lymph, they retained in our blood or serum for a longer period of time. So whatever immunization we're talking about, we're always talking about the immunoglobulin G. That could be transferred from one animal to the another animal, could be transferred from one animal to the human being. So that's the way we are getting the immunization. So say, for example, I will talk about the vaccine in some class later. So somebody got bitten by a poisonous snake. So So you are going into the health center or hospital. So they're giving some anti-venom. Okay. So anti-venom is nothing but an antibody produced against that venom. Okay. So what class of antibody? It's immunoglobulin G. Okay. And as they found 80%, so relative abundance is quite high and also specific, very much specific. So immunoglobulin G is used for immunological research and clinical diagnostics mostly, unless you are specifically interested to know about other antibodies. Now, this human IgG could be subdivided into 4 different subclass: 1, 2, 3, 4. There is no much meaning about these numbers. There is a slight difference in the gamma chain sequence, and another one is which one is found in which concentration in the serum. IgG1 is about 60% or more than 60%. IgG2 is found 20% to 30% range. This is even less than 10%, and this is even less than 4%. Okay, so we do not much sure about the role the IgG3 and IgG4 plays, but we know about the IgG1 and 2. IgG1 is actually found to be responsible for immune response against polypeptide or protein antigens. Okay, and most of the time the antigens are polypeptide or protein in nature. IgG2, on the other hand, is effective against carbohydrate or polysaccharide type of antigen. Okay, and IgG3 is also found to be protective against polysaccharide antigen, but we do not know the exact role they play because we already have IgG1 for the same type of function. And the IgG4 that is found in the later period of time that found to be bind to the polysaccharide antigen, but clear role is not known to us yet. Okay, so that is all I have for today. So I will be stopping at this point for today's class. So now, if you are there, if you have any questions about this antibody and antigen, what we have discussed today, this is the time you can ask me. Is there any question? Okay, if you do not have any question, that is also okay. Then I have to say formally, maybe you already know, so that you have to appear for the next round of exam that is called minor test 2, and that will be happen on that will take place on the next Tuesday at 2:30, not Wednesday. Be it clear, because Wednesday is a holiday on account of the Mohalaya. So 2:30. And all other procedures of the examination will remain same as it is on the minor test 1. So it will be a 25 marks post examination. You'll be given 45 minutes of time. Questions will be multiple choice in nature. And I will produce a question paper to the MS team. I will provide a link, maybe 5-10 minutes prior, but that will be effective only by 2:30 or 2:45. I will let you know through WhatsApp or email, whatever. So see you on the 2:30 on the 5th October. Do you have any questions in this regard also? Exam?
No, sir.
Okay.
Sir, how many questions will be there?
25. 35 marks, 25 questions, 45-minute times. Okay.
Okay, sir.
Okay.